Hello everybody, welcome back to another episode of STEM Nerds. I'm Jason. Glad you're here today because in today's video we have got to talk about genetic mutations and the way that organisms, in particular here we're going to focus a little bit on bacteria, how bacteria exchange genetic information and how they acquire new genetic information, because that can lead to antibiotic resistance. Thanks for being here. All right, so first things first, we have to go over a few terms just to make sure we are all on the same page when it comes to the language that we're using to describe these mutations. First thing, when we're talking about the genotype of an organism, we're talking about the genetic makeup of that organism. So when we're specifically looking at mutations, the mutations could be potentially changing the genotype. That in turn can lead to changes in phenotype, the physical characteristics of the organism. When we look at the genetic makeup of an organism, they can either be haploid or diploid. So as an example, our prokaryotic organisms, bacteria, are haploid. They have one copy of each gene. Human beings, on the other hand, are an example of a diploid organism. We have two sets of genes, one coming from each parent. Now that matters because if there is a genetic mutation that happens in an organism that is diploid, there might be the correct version of the gene still there able to function. Now, if you ever hear an organism described as a wild type organism, that doesn't mean it's crazy. It just means that the wild type organism is the one that is found predominantly in nature, as opposed to a mutant that has obtained a genetic mutation that could lead to a phenotypic change, and that's typically going to be less common. Finally, we have the auxotroph, and an auxotroph is a very specific type of mutant, and this mutant requires additional growth factors that it used to be able to make on its own before a genetic mutation. So organisms and their populations can change genetically in different ways. One of the most common is natural selection. Organisms best suited for an environment are going to leave behind more offspring, and therefore the genotypes of those organisms are going to be best suited for the environment at that time, but genotypes can change depending on whether or not the environment changes. Organisms can also undergo mutations, changes in the genetic code that can lead to changes in phenotype. And finally, we have genetic transfer, and this is especially important in bacteria that are able to exchange genetic information and acquire new genetic information from other organisms or once living organisms. So here's an example of what gene transfer looks like. In particular, in bacteria, we have two different types. We have vertical and horizontal gene transfer. In vertical gene transfer, what's happening is the organism has a particular genome, and every time that organism grows and divides, it leaves behind more offspring, pretty much just like it. In this particular example, we're dealing with bacteria that undergo binary fission. They make a copy of their single circular chromosome, and all the offspring, in theory, should have the exact same genetic makeup. If a mutation occurs at any time, the subsequent offspring will also have that genetic mutation. Horizontal gene transfer is a little bit different. In this particular case, what's happening is what is known as bacterial conjugation, and in this case, one microbe can exchange genetic material, or actually provide genetic material, to a second microbe. What's interesting is after that recipient organism has received that new genetic information, it can then also go through vertical gene transfer, make a lot more cells, and those new cells will also have that new genetic information. So I want to turn our attention here. We're looking at different strands of DNA. The top strand, well, that strand of DNA is what we're going to call the wild type strand. And then you're going to see we have 4 additional examples, and I've highlighted in red for you that there's been a change in the genetic code. In other words, there's been a mutation. It turns out that there are different types of mutations, and these different types of mutations can have different outcomes. And so what our job is, what your job is, and this is great practice for you, is we want to take that DNA, transcribe it into RNA, and then utilizing this table, translate that RNA into a series of amino acids. So actually, I think it would be a really good idea for you to take a few minutes, pause this video, and do that. And then we're going to come back and we're going to see these different types of mutations. All right, so here we've got the first example. This was the wild type example. This is the RNA. That is the result of that single strand of DNA that I showed you, that first line. And so now we have the RNA here, and we took that RNA, and you can double check yourself, and we have an amino acid sequence down below, methionine, tyrosine, histidine, glycine, and alanine. And this is our wild type. This is what we're going to be comparing all of those other genomes to. Okay, so again, wild type is at the top. Now we need to focus on that second line where we have that highlighted T and see what that has done to our genetic code. What has it done to the amino acid sequence? All right, now the second one that we have here, we can see we have that highlighted T, and we can see the change in the RNA. We had the AUG, the methionine or the start site, but then the very next codon, look what happens. We have UAA, and if you said UAA equals a stop codon, you're 100% correct. At that point, we don't have any more translation happening. And so when you have a genetic mutation that leads to a stop codon, we call that a nonsense mutation. And here we go again. And so the first one was a wild type. The second one, that was our nonsense mutation. Let's go ahead and take a look at the third one with that highlighted G. All right, so now we've got that G highlighted, and you can see the subsequent change in the RNA sequence. We have a C there. And now what's going on is we have methionine, tyrosine, but we have histidine here. And that histidine is the exact same amino acid we had originally. And therefore, when you have a genetic mutation that leads to the same amino acid being inserted, well, we call that a silent mutation. Now remember, we talked about this in a previous video. Just because we have the same amino acid doesn't necessarily mean we have the exact same three-dimensional structure of a protein, because that ribosome might have slowed down a little bit to incorporate that histidine, and therefore that protein could be folding just a little bit differently. Okay, let's see what happens. We have that C highlighted now. What is going to be the change? Well, here we have that highlighted C, and now we have CAG. That is our codon, and we can see here our RNA has changed, and now our amino acid sequence has changed as well. And so that third amino acid is now different. When you have a genetic mutation that leads to a change in the amino acid sequence, we call that a missense mutation. Now, this could be bad or somewhat indifferent. If the amino acid that was inserted had the same general properties, like the R group was pretty much the same, we might not have a protein that folds too differently. Now, there are some instances where a single point mutation like this can have a massive difference. Example being sickle cell anemia. But again, the point here, when you have an amino acid sequence that is different from the original, from the wild type, because of a point mutation, that is called a missense mutation. Now you're going to see this last one is a little bit different because we have something going on here that we haven't seen before, and that is an insertion. That A has been added to the DNA. Let's see what happens to the RNA and then the subsequent amino acid sequence. And so here what we've got is what is known as an addition or a deletion, insertion or deletion. That leads to a frame shift. Now what's happened here is every single amino acid from that point on is probably going to be different. This almost always leads to a messed up protein, one that is going to be very different than what was intended for the wild type version. A lot of times you're either missing stop codons or you're inserting one anyway. And so this is usually going to be the worst possible scenario when you have an addition or a deletion that leads to a frame shift. So can I just give you a really interesting example? So I have these concepts here about frame shift, removal, and addition of nucleotides. What's on the screen in and of itself, the words here are not the important thing, but we can read these words, right? We understand the message. In a frame shift, let me show you what's happening to a ribosome, because just like us, ribosomes, as we've already talked about, read in codons. They read in sets of 3. But if you add or remove a nucleotide, you get a very different message. And so look at this. Here's what I've done here. I removed the R, and so it was a deletion. But I left the spacing exactly the same. Does this make sense to you? Probably not. And that's exactly what's happening to the ribosome. That's exactly what's happening to the subsequent amino acid sequence and protein that is manufactured as a result. We've got basically garbage. And so these frame shifts, additions, and deletions are a really bad thing. And you can see that here because the message gets all screwed up. Okay, so let's summarize this. Let's try to make it easy. Let's get all of our thoughts together when it comes to genetic mutations. Everything we've talked about so far is a point mutation. A single point within the genetic code has been altered in some way. From there, we have two main categories. We have the base substitutions, or we have additions or deletions, so one or the other. If it's an addition or deletion, like we just talked about, that leads to a frame shift, almost always going to lead to a useless protein. The base substitutions, well, we have three potential outcomes. A silent mutation, which we already learned, means that we have a mutation, to be sure, in the genetic code, but the subsequent amino acid is the same. We have the missense mutation, where that change in the genetic code leads to a different amino acid. And then we have the nonsense mutation, where the difference in the code, that point mutation, leads to a stop codon instead of an amino acid. Now, in addition to the point mutations we've already talked about, there are other types of mutations. There are things like transposons or jumping genes, where parts of a genome will remove itself from one location and find a home in a different location. That can be really bad, knock out a gene. And then we've got things like this, a thymine dimer. And so this is an example of what happens with UV radiation. If you go out into the sun for a long time, the UV radiation can hit our cells, hit the genetic code, and cause what are known as thymine dimers. So anytime there are two T's next to one another, like we're seeing on the right-hand side, they can covalently bond to one another. And that makes it really hard for the polymerases, DNA and RNA polymerase, to interpret the message, because the message doesn't really make a whole lot of sense if these mutations happen in areas where they are controlling cell growth. Maybe this is a gene that controls cell growth, and that gene is no longer functioning. That can lead to certain types of cancers. Now, there are mechanisms to fix these genetic mutations. Both prokaryotic and eukaryotic organisms have what is known as proofreading ability, where DNA polymerase, the enzyme responsible for replicating DNA, something we've talked about in a previous video, that enzyme has proofreading ability. At the time, I said, when we were discussing replication, I'll put that video in the upper right-hand corner, we said that when we either have two purines or two pyrimidines bind, or the wrong purine and pyrimidine bound together, that creates a discontinuity in the shape and the structure of the genetic material, and the DNA polymerase can often sense that and correct it. If a mutation happens after the fact, like what we're talking about with the thymine dimers, or for some other reason, chemical mutagens, this is where excision repair comes in. And excision repair requires a few enzymes. It's going to require an endonuclease. And so these endonucleases, what they do is it's basically like a little pair of scissors that will not only remove the single point mutation, but a few bases upstream and downstream, and kind of just cut that part out. Then we'll have a DNA polymerase come in and redo it, correct it, and then ligase will seal up the gaps, because we already learned that DNA polymerase can't do that. The same general thing can happen with thymine dimers. That can either happen in visible light or in the dark. And in our microbiology classes, we actually do an experiment with a microbe that uses visible light and enzymes to repair thymine dimers. There are also dark repair mechanisms that rely more exclusively on excision repair to correct when those thymines are connected together covalently. Bacteria have an additional defense mechanism. It's called the SOS system. And this is when DNA is heavily damaged. There is a series of enzymes that come in and do the best job that they can of repairing either double-stranded DNA breaks or large chunks of DNA that have been missing. This is a system that, again, it's prokaryotic in nature. In eukaryotes, this would not work out so well, because think about it. We are multicellular organisms. If we had a liver cell, let's say, that was not doing well and it had a bunch of problems in the DNA, and we try to fix it, and using the SOS system, and all of a sudden the cell's like, wow, you know what? Bunch of mutations here. I'm just going to grow out of control. That would not be good for a multicellular organism. For a single cell, it doesn't really matter, but for us it does. And so there's really not a point to having an SOS system when you can just, that cell can die and the other surrounding cells can replace it and fix that tissue. Now, at times, us human beings, we want to induce mutations, and usually the purpose for inducing mutations is to discover the purpose of a gene. What do genes do? How do we figure that out? Well, sometimes the way we figure it out is we take a model organism like bacteria, yeast, or an earthworm, and we knock out a gene. We purposely mutate it. And then we look at the organism and say, okay, well, what went wrong? And if we can figure out what went wrong, we know, okay, the gene that we mutated does this because that's the thing that's broken now. Now, there's lots of different ways to induce mutations. You can do it chemically with base analogs. Think of a base analog as like a fake base. It's like a fake A, T, C, or G. These base analogs are actually really important as antiviral medications because viruses insert so many bases, and when they get these analogs, the DNA polymerase, the RNA polymerase, they don't know what to do with these things. They can't interpret that message because it's not the real message. It would be like if you only speak one language, and then every once in a while in a word, I start throwing in symbols and letters from a completely different language and you have no idea what it means. And so that's what's happening with the base analogs. The intercalating agents, what they do is they kind of pry themselves into the DNA, and that will often cause additions and frame shift mutations. And of course, there's transposition. We already mentioned this as a jumping gene, but you can do this on purpose. You can actually take parts of genetic information and plug them into a fully functioning gene to knock down, which means to slow down the function of that gene, or knock it out, which means to stop the function of that gene. And of course, radiation can be used as well. This is why we use it in healthcare, because UV radiation is non-ionizing, causes those thiamine dimers, but that can destroy bacteria, which is a good thing. Obviously, if human beings get too much of it, that might not be a good thing in terms of cancer. So we've got the UV, non-ionizing radiation that can induce mutations, as will X-rays. That is an ionizing type, higher energy, does a little bit more damage. But again, the main point is if we are inducing mutations, usually it's to determine what a gene actually does. So if we introduce new genetic material or we cause a mutation, how would we know that we've actually done it? Well, we have to select for the microbes or the organisms that are mutated. And there's two main ways. One is called direct selection. I'm going to use an example that we have done many times in our lab. It's called the pea-glow experiment. And in the pea-glow experiment, what we're trying to do is we're trying to introduce a plasmid. Remember, plasmids are extra chromosomal pieces of DNA, not necessary under normal conditions, but this plasmid has an antibiotic resistance gene, and it also allows microbes to glow green under UV light. And so what we're trying to do is we're trying to get this plasmid into the cell, and so we're altering the genetic information of that cell. Well, how would we know that we did that? For pea-glow, it's a process called direct selection. We take the microbes after we've exposed them to the plasmids, and we put them on a nutrient agar plate, which is what you're looking at here, and we see if they grow. Because these plates have ampicillin in the food, in the environment, and they also have another component that will allow them to glow green if they take that component in. And so as long as we've transformed the microbes, as long as we've gotten that plasmid into the cell, then we're going to see it on the plate. That is direct selection. We're selecting for the microbes that have the genetic mutation. They're either going to be there or they're not. Sometimes in our lab, we are not successful and we don't get any growth on these plates. But we know the ones that are there have been genetically altered. Now it gets a little bit more complicated when we are dealing with an auxotroph, and these can be really useful. So, if you remember, auxotrophs, these are organisms that experience a genetic mutation, and that genetic mutation does not allow them to produce a growth factor that they used to be able to produce. Example, let's say we've got some microbes and we are going to blast them full of UV light. Now, UV light, as we know, causes thiamine dimers. It can cause genetic mutation. If we take those microbes and we grow them on complex media that has all the growth factors they're going to need, whether we've mutated them or not, they should grow. So how are we going to know if we actually successfully mutated any of the microbes? Well, step 2, right there in the middle, we can actually take a piece of filter paper or a piece of velvet and kind of press it down on the plate, almost get like a fingerprint of the plate, and then press it on 2 new plates. And what we have is 2 different environments. On the left-hand side, we have the same media that we used originally, complex media. It has all the stuff that the microbes need in order to grow. But on the right-hand side, we have what's known as a minimal media. And in that minimal media, it's missing a particular growth factor. It might be one of the essential amino acids, usually is. And so what we see here is a comparison between the microbes that are growing. Now, usually when you're mutating these bacteria, each one of those little orange circles represents an entire colony. That might be up to 1 million or more cells in that single dot. So what we're looking for on these two plates, these nutrient agar plates, the complex media versus the minimal, is the growth patterns. Anytime there is a colony missing from the plate on the right-hand side in the minimal media that is present on the left-hand side in the complex media, we know that that colony is actually comprised of auxotrophs. So as long as we give the microbes everything they need to grow, they grow just fine on the left-hand side. The minute we take that growth factor away, they're not growing. And so that missing colony, where that little line is that says auxotroph, that's on the left, it's not on the right, we've identified it. Why do we do that? Well, we can use the auxotrophs in experiments in really valuable ways. And so we identify that auxotroph. Now let's go to the next step, because the next step is where it gets really useful. So what we have here is an example of the Ames test. And the Ames test is designed to determine whether or not a chemical is a potential genetic mutagen. Well, we don't really want to experiment on multicellular organisms, especially ones that are cute and fluffy, and so we start out using bacteria. Now, at the top of the picture here, we're going to see that we have our auxotroph. That auxotroph was the colony that we identified in the previous slide. This auxotroph is tryptophan deficient. In other words, if you give it tryptophan in the media, it will grow just fine. You take it away, it cannot make it on its own, it will not grow. Okay, so now, next step is we're going to introduce that suspected mutagen. That's the tube on the right-hand side. And we're going to put that in the media, right? It's minimal media. The media itself is tryptophan deficient. And so what we should be seeing are microbes that are not growing in this media because they can't make the tryptophan. We're not giving it to them. And therefore, what we should be seeing is the plate on the left. Yeah, there's always going to be a little bit of natural reversion where the mutation that was induced via maybe UV radiation that we did earlier reverse mutates on its own. Really, those two dots represent 2 cells. Out of all the microbes that would be in that original tube at the top, 2 cells went back and reverted back to wild type. That is next to nothing. Where we get concerned is if we see the results on the right where it says probable mutagen. If we have a situation where we have a lot of cells that are reverse mutating back to wild type, now that's good for the bacteria, but that is not a good sign for our suspected mutagen. That means that that chemical is most likely interfering with DNA and reversing the mutation we did originally. We got to be really careful with that. Definitely warrants further study. And so the Ames test, its purpose is to determine whether or not a suspected mutagen actually interferes with genetic information. In this case, what's happening is we're getting a reverse mutation if the suspected mutagen is actually interfering with genetic information. Last concept we're going to talk about is there are other ways that microbes can acquire new genetic information, and that is through a process called conjugation. Here, and so basically what we have 2 cells at the top, and we label these cells F plus and F minus, and the F stands for fertility factor. So the F plus cell is going to initiate the contact between the 2 cells. The F plus cell in the upper left-hand corner, that is our donor cell. The F minus cell does not have the ability to initiate contact between 2 cells, and so that is going to be the recipient. So this F plus cell, thing number 1 is it has an F plasmid. And this plasmid carries information for the F plus cell to make that pilus and be able to transfer the plasmid from itself to the recipient on the right-hand side. And so we see that process as it plays out. What's happening is the donor cell on the left is making a copy of one of the strands of the DNA, transferring it to the recipient on the right-hand side, and the recipient will make the other complementary copy of that plasmid. In the end, what's important to understand is now both my donor on the left and my recipient on the right are both F plus. Going back to earlier in the video, this is an example of horizontal gene transfer. This can be a big problem when it comes to antibiotic resistance because Often, these F plasmids also carry genetic information that allows a cell to do other things besides just make this connection and transfer plasmids. A lot of times there are antibiotic resistance genes on these plasmids, and now the recipient is also antibiotic resistant. And don't forget, once these cells have those plasmids, through vertical gene transfer, they can have lots and lots of babies, and all of those offspring will also have those plasmids that are antibiotic resistant. Now, something else to consider, there is a variant of this process, and it's called HFR, and that HFR stands for high frequency of recombination. Now, I don't have a real pretty picture for you, so we're going to have to use our imaginations a little bit, but imagine the F plus cell on the left-hand side, instead of having that plasmid be extra chromosomal outside the chromosome, that orange plasmid is actually part of the blue chromosome. Now, when that happens, that has been the plasmid and the chromosome have been recombined, and thus the HFR, high frequency of recombination. And so that F plus cell would actually be labeled HFR. Now, it can still do all the things an F plus cell can do in terms of making the pilus, making the connection, and attempting to transfer genetic information to a recipient. The only difference is you can see that there is an origin of replication, that little dot on the fourth cell down on the left-hand side. Well, what's happening is because the plasmid is stuck into the blue chromosome, you have the origin of replication about halfway through the plasmid. As replication happens, as that transfer happens, we're getting plasmid, but then we start going into the blue chromosome. The connection is not going to last nearly long enough to get all the way around the blue chromosome and back into the rest of the orange plasmid. So what winds up happening is we see, okay, we've got origin of replication, orange plasmid, we get into the blue chromosome, a little bit of that gets transferred over, the connection breaks. The recipient cell, when it's connected to an HFR, is going to remain F minus because it really only got about half the plasmid, a little bit of the blue chromosome, but it really doesn't have the ability to transfer that information to any other cell. And that can actually be a good thing sometimes. Now, if it did receive that half of plasmid, that allows it to be antibiotic resistant or gives it some other special ability, that could be an advantage to the F minus cell, but the F minus cell cannot transfer that information, at least not horizontally, to any other cell. It remains F minus. All right, so we talked about the middle one, conjugation. There's two other ways in which cells can transfer information. One of them we've kind of alluded to already, and that was that pea-glow experiment earlier, and I used this word called transformation. And the transformation is kind of interesting because a cell on the left-hand side, top left-hand side, is going to be our donor. But in transformation, what's happening is usually that cell is going to be lysed. So it's dying for some reason, the cell lyses, the genetic information is released from that cell, and nearby cells that are naturally competent can bring in that genetic information and incorporate it into its own chromosome. And there might be new genetic information there that provides an advantage to the recipient cell. And so the cell itself on the right-hand side has to be what we call naturally competent. It has to have the ability to take in DNA from the environment. Cell on the left, unfortunately, is dead. So our donor cell is dead, but our recipient cell might receive new genetic information. There are ways that we can make cells competent, but do so artificially. And so in a lab, like when we're working with pea-glow, we use things like calcium chloride. So we can use chemicals. We can use heat to ensure, or at least to increase our success of artificial competence and transformation. Electroporation, using electrical current. Sonication, using sound waves. Those things can all be used to make a cell that is not naturally competent capable of bringing in genetic information. Now, last one, and that's at the bottom here, that's transduction. This is an interesting concept. What happens is a bacterial cell gets infected by a virus. Viruses, when they enter cells, usually take that cell over, especially in bacteria, chop up the bacterial chromosome, and as the virus is replicating, it's incorporating its genome into its capsid, that little head region of that virus, and then it leaves and goes and infects another cell. But some of the viruses, because they're not very bright and they lack a brain, they don't incorporate their own genetic information into their capsid, into that little brain-looking area. Instead, what they do is they incorporate bacterial genetic information into the capsid. Then when they go and infect a new cell, like the virus is doing here on the lower right-hand side, it actually injects bacterial genetic information and not viral. And when that happens, the recipient is not infected by a virus. It's just got genetic information from a cell that was previously infected by a virus that is no longer there. And so if we were to summarize, in both transformation and transduction, the donor cell winds up dying in the process. It's only in conjugation that the donor cell is alive after the transfer of the genetic information. All right, everyone, so that was a decently quick overview of genetic mutations, why we induce them, how cells can fix them, and how cells, in particular bacteria, can transfer genetic information from one cell to another. As always, thank you so much for being here. Have a great week, everyone.